I'll be presenting a practical approach to intestinal permeability techniques that support biopharmaceutics classification system. This is the outline. I'll spend a few minutes discussing the extendability of BCS beyond biowaivers. There is so much more you could do with this tool in terms of a developability context, as the speaker before me said, risk management context. The majority of my presentation will discuss permeation techniques, will discuss our experiences and delineate considerations and solutions that we have found along the way. I'll be also quickly comparing the attributes across these techniques, attributes including range, variability, sensitivity, and how certain times techniques converge to find a common solution. And finally, provide some conclusions both from the producer, industry perspective, and also from the consumer perspective. And then we will try this in with an economical sense. So real brief, as was just stated before. BCS is a scientific framework based on two simple principles, aqueous solubility and intestinal permeability. So when you combine these with drug dissolution, you have the three major factors that define the rate and extent of drug absorption. When each of these factors cease to be rate limiting, as in when you have high solubility, high permeability, and rapid dissolution, that's when a biowaiver is possible. So there are many different ways you can define a biowaiver. One way of looking at it is regulatory acceptance of in vitro testing as a surrogate for an in vivo BE study. This image is from the FDA's individual product recommendation that basically states that for this compound, the BCS waiver option is an acceptable and a suitable alternative to establish therapeutic equivalence. But as mentioned before, there is so much more you could do with an early classification. Of course, the biowaivers, but there is a possibility of predicting drug disposition in relation to both metabolism and transporters. The possibility of predicting food effects to set up a guide for your salt and polymorph selection, to hone in your formulation strategy. And in this slide here, if you look at the top right panel, these are the class 1 drugs. Most times your best behaved development compounds. So as you can see in this formulation, if you're going after oral drug delivery, not as many major challenges. Food effects, mostly none. Metabolism is significant first pass, as in the compounds have very high permeation, get into the hepatocytes, have easy access to drug metabolizing enzymes. Transporters, even though these interactions exist, since you have high solubility and a high rate of transfer, you have the ability to overwhelm transporter interactions. So as you look at these different classes, you can see that class 1 has the least amount of language in here. Everything else is much more dense, essentially indicating that there is much more work to be done. Development pathways for other classes, much more intricate. So moving on to permeation techniques. I'll be talking about 3 different techniques. Excise tissue. This is human and animal intestinal tissue. We'll be discussing rat intestinal perfusion and also a monolayer of cultured epithelial cells. Before getting into the specifics, I wanted to give you a perspective in terms of acceptance, both from the industry side and also from the regulator side. So this is for one technique, in this case KECO2, also for one specific application, in this case classification or biowaivers. As you can see, there is clearly an adaptation curve. The bar graphs here is the number of studies that we perform as a company, and as you can see from about 2008, we are running at about 30 studies per year for a combination of both classification and biowaivers. And this is specifically what the OGD has seen for biowaiver requests. So many of these, of course, they take the next step for submission and for approval. So both with the Office of Clinical Pharmacology and the OGD, numerous compounds come in both from the innovator side and from the generic side for biowaivers. Myriad techniques can be used, clinical and non-clinical, and that's what this pie chart depicts. clinical techniques, mass balance, absolute BA, and in terms of non-clinical, majority of the studies apparently use KCO2. There's been only one submission that we are aware of which used rat perfusion. So what makes KCO2 in a sense more reliable when compared to these other techniques? Before I get into those details, just a little bit of catch up where we talk about what exactly are these cells, how do you perform these assays, and then we get into considerations and solutions to essentially create a robust, validated technique. So these cells are derived, they're human-derived, they're colorectal adenocarcinoma cell line, they're continuous as an immortal, and when grown under specific conditions on a polycarbonate filter, for instance, they become confluent, they polarize, they differentiate, and they start resembling the human intestinal enterocyte. Morphology, they both share a very similar cobblestone, that was not me, cobblestone type of structure. And as you can also see that in terms of enzyme expression, there are many uptake and efflux transporters that are also present in these cell lines. Most times, the assay format is bidirectional permeability. You would run this in the apical to basolateral direction for classification purposes. This is when you would compare the rate of transfer to that of a high permeability internal standard, and you also evaluate in the basolateral direction to confirm that there is no asymmetry, because the current guidelines require that for utility of in vitro test systems, you have to be passively diffused. This is the equation for determination of permeability. Terms to note here is your initial rate of the permeability time course profile, and also in the denominator, the concentration of free drug that is available for diffusion. And this is how the efflux ratio is computed, which is essentially permeability in the B to A direction over permeability in the A to B direction. So moving on to considerations, things that we have learned along the way as we've been running this now for about 11 years for classification, for biowaivers, and various other applications. One of the first things we learned real quick was having the right number of time points, having the right assay duration is very critical, because if you have a compound that has a high rate of transfer, at the later time points, there is already a tremendous amount of depletion from the donor side. What that does is you start seeing this plateau on the receiver side, and if every time point was chosen for permeability determination, you would underestimate the value. The corollary to that is for a low permeability compound. The first few time points, you might never rise above the limit of quantitation, and you almost need these later time points to plot the slope and compute permeability. Another consideration is stability. So this is where it is helpful to have the system validated under multiple donor pH conditions. More recently, after this open forum on BCS, there has been discussion on local and average permeability. Certain times, if you just assume local permeability, you could still get misclassification on the conservative side, where erroneously you might say your compound is not class 1, and hence the concept of average permeability. So in this case, compound is tested using a pH 7.4. Recovery is not where it needed to be. Usually we look at 80% as our cutoff, and then you realize that this is because the compound is not as stable at pH 7.4. In this specific instance, it did not matter, because the Tmax for this compound was about 1 hour, which is more consistent with a median intestinal pH of maybe 6.5 or 6.8. So the biowaiver study was essentially performed using 6.5 on the donor side. We get much better recovery, and biowaiver is accepted. Another consideration, and this is not necessarily for a biowaiver, this is more for classification purposes, so that you're able to currently rank class 2 versus class 4, that there is sufficient amount of compound on the donor side to get an accurate estimate on the receiver side. So when confronted with low solubility, you might have to use certain excipients, and again it is critical that you evaluate these excipients in the sense that they do not impact cell line integrity, that they do not impact rank order of your control compounds, and that they also do not impact transporter expression. So once you build this list of qualified excipients, you now have a tool that can do much more than just biowaivers. Another consideration is non-specific binding, and this is again very similar to the excellent example of essentially the stomach not having a paddle. So very similar to that. When you run these in transfer apparatus, there are all these different places that the compound can stick to, on the insert, on the well, to the filter. So these are all artifacts of running an in vitro test system, and unless you have measures or techniques or procedures to alleviate that, you might end up with erroneous conclusion of poor recovery, so indicating that the compound might be unstable, so the possibility of a second form in the lumen, and do we have to go investigate that? All of that untrue, just being an artifact of the in vitro test system. So measures you could use to relieve that, the possibility of adding BSA on the receiver side, this makes it more physiological. So when the compound traverses through, instead of sticking to the apparatus, BSA keeps it suspended and also maintains sink conditions. You could pre-incubate certain times, load the cells so that your recovery is enhanced. And when you ultimately calculate recovery, not just go after the donor and the receiver compartment, but also lyse the cells so that you have every compartment accounted for. This basically illustrates that BSA still has a similar rank order and good correlation when run without BSA. An example of mass balance, the compound had good chemical stability. Recovery was not as great when we only measured the donor and the receiver compartment. Again, the possibility of erroneously concluding that this is unstable, but when we go back and track it down in every possible compartment, we are able to get this number past the 80% threshold. Another consideration is tolerability. So Keiko2 cell lines is a monolayer on a filter. And sometimes the concentration of compound that it can tolerate, the concentration of excipients it can tolerate, might be different from that of an in vivo situation where you have a multi-layered tissue. So biowaivers require that you run 100%, 10%, and 1% of the highest dose strength. And sometimes this is just not possible because at the 100% of the highest dose strength, the cell integrity is compromised, and essentially you have a leaky monolayer. So in those cases, so this is an example where the 100% did not work, so we come back and start titrating the concentrations down to find what is acceptable. And also the belief that the lowest concentration is the most critical, because at the lowest concentration, you have the greatest possibility of exhibiting asymmetry. And as long as we have the lowest concentration that does not exhibit asymmetry, and we have a range, we believe that this is sufficient to satisfy the criteria of choosing a range of concentrations. It does not always have to be 1, 10, and 100%. And we have at least 2 successful biowaivers using this truncated range because of the insufficiency of test system at the highest concentration. This is an important one. What is the ruler that you would use to essentially classify your compound? Numerous suggestions. Back in 2000, the FDA's recommendations was to either consider antipyrin or metoprolol. Antipyrin has permeability of about 63, and it is the highest compound in maybe the 150 that we have assessed. So that would not make sense. Metoprolol was right about the 95th percentile. Again, not a great idea because the spirit of the guidance is to choose something at the proximity of the low-high class boundary. So we decided that maybe we'd choose Pindalol. More recently, FDA perspective has been considered Labetalol. But again, Labetalol is a PGP substrate. There are differences in its permeability between 6.5 and 7.4. So this compound, which from in vivo, is clearly class 1, greater than 90% absorbed. Now that does not always correlate to a high rate of transfer, but it is possible. So what we recognize that using Pindalol, we did not, there was a conflict between the in vitro and the in vivo result. And this was not the first instance we had seen this. We had seen other instances where data was just not correlating. So it made us think that maybe there is a better HPIS at the proximity of the low-high class boundary. So as we continued with our investigation, we realized that maybe this compound, minoxidil, which has a permeability around 4 or 5, is actually a better marker of this low-high class boundary, where there is still about a 5 to 7-fold separation between the highest low and the lowest high. So this is the compound that we now use for our biowaiver classification. And if your compound C had a P apparent of 11.8, that is easily higher than the current HPIs, and several compounds of the past 3 or 4 years have been successfully biowaived using this internal standard. So this is the data from about 110 compounds. Essentially, the x-axis here is CACO2. The marker here is minoxidil. The y-axis is fraction absorbed. Again, the marker is 90%, so data can be divided into 4 quadrants. Quadrant 1 and 3 is where there is agreement between in vitro and in vivo. This is for about 86% of these 110 compounds. Quadrant 4 is where the rate of transfer is low, but in vivo absorption or complete absorption is possible, is high, which is not surprising because rate of transfer is one of the parameters that defines extent of absorption. The other parameter is the residence time. So there are examples such as sotalol and practilol possibly, where differences in the intercellular junctions of CACO2 and also its reduced surface area compared to the upper GI would make this maybe a less successful model. Also, sometimes uptake transporters are not expressed to the extent that they need to be, and that would be examples such as pregabalin and such. So this is understood, and this is a producer risk, but never a consumer risk, because CACO2 always errs on the conservative side. Now these compounds here are where the 5 data points, every one of these is a highly variable drug. And in vivo data for this is either very variable or ambiguous. And when you run these simple in vitro experiments such as permeability, solubility, dissolution, these compounds easily turn out to be class 1. And in every one of these cases, the current individual product recommendation put forward by the FDA lists the BCS biowaiver as a feasible option. So this is again instances where in vitro provides clarity which might sometimes be lacking with just in vivo data. So you might have to look at more than one approach. So moving on to the next technique, excise tissue. Not only can the format that you would use for intestinal permeability can also be used for a variety of other tissues, such as dermal, ocular, nasal, vaginal. Essentially, what limits you is availability of tissue, and then going through the validation to make sure that you're able to set up the model. The way these studies are performed, tissues obtained post-mortem, and rinsed, cleaned up, dissected into small pieces, stripped of the underlying muscle layer, and mounted in these Assay chambers, where essentially what separates chamber A from chamber B is this excise tissue. You will have to have set up criteria for tissue viability and integrity so that your experiment is initiated and completed within that assay window. Considerations and solutions very similar to permeability as far as solubility, stability, non-specific binding goes. There are some unique challenges. Tissue thickness, it is no longer a monolayer. So in addition to sampling on the receiver side to compute P apparent, we also look at disappearance from the donor, because sometimes your compound might never make it across to the receiver side. And again, we have ways of making sure that mass balance is not a problem, and variability is larger than what we see in a CACO2 cell line. So what has worked better for us is to use the ratio with a co-dosed internal standard as opposed to just the absolute P apparent number. So this orange line here is essentially the ratio, which flattens out. But if you look at the P apparent, there's much more variability across the different segments and such. And then you build an acceptance criteria, which is essentially a minimum of 2-fold separation is required for each replicate to be considered successful. Correlation against versus human fraction absorbed. Different species, human jejunum versus human fraction absorbed. Rat jejunum versus human fraction absorbed. Non-human primate jejunum versus human fraction absorbed. Again, if you have the ability to mount the tissue You can go through a validation, and most likely the correlation will make sense. In addition to this, correlation with fraction absorbed, these tissue studies can also be used to look at transporter interactions. This is where compound B is a peptide-1 substrate. So when challenged with glycine, there is a tremendous decrease in its P apparent. You could also study impact of excipients where we're looking at EGT and sodium caprate. They're expected to compromise or open up the tight junction space. So as expected, the permeability of atenolol increases, antipyrine does not change. The next technique is intestinal perfusion. There's two ways you could run this, closed loop or open loop. Closed loop is a static model where your dosing solution is in contact with the intestinal tissue for the entire duration. With open loop, either you can go single pass or recirculating. Both of these are similar and equally popular type of models. In addition to monitoring disappearance from the donor side, you could also pick up the compound that appears on the plasma or the serosal side. So you could look at this in the portal vein, in the mesentery, femoral, multiple options. Again, considerations and solutions. The first one is stability, because here stability or disappearance from the lumen is indicative of permeability. So instability is a parallel and a competing process, and essentially that would result in your C out being underestimated, your P effective being overestimated. So this is a much bigger challenge in intestinal perfusion, that you have to make sure your compound is stable in SGF and SIF or the media used for these assays. When you think about KCO2, that is predicated on compound appearing on the receiver side. So if there is instability, again, it becomes a producer risk. Whereas in this case, P effective is overestimated, so that becomes more of a consumer risk. Solubility. Again, when you have compounds that have low solubility, there is always the potential misclassification, class 2 versus class 4. So what can be done about it? Of course, you can start thinking about excipients that can be used. But as discussed before, these excipients have to be carefully selected. You have to make sure that they're validated, they're suitable, and they do not actually impact your permeation characteristics, because that also compounds the risk of misclassification. So the desirable impact here would essentially be if you had the ability to always correctly classify, because a class 2 will have fewer SUPAC constraints up to level 2 changes. With a class 2, you're also able to use in vitro dissolution, maybe to waive some biostudies based on proportional similarity and such. And recognizing this, Absorption Systems is actually now working with a major pharmaceutical company, because we believe that this problem of misclassification is much more prevalent industry-wide. And if there are members in the audience that have similar interests in this effort, I would welcome a discussion just to see what thoughts you have about the risk of misclassification between class 2 and class 4. Non-absorbable marker. This is where it is not working as well, because as you can see, atenolol data not only bounces around, but it's also a negative number. And these markers have been used since the inception of this technique, and we believe that it's probably time to bring some 21st century innovation to choosing a better non-absorbable marker, and that's an effort that we're also currently working on. And to solve problems of sensitivity, because sometimes when you have very high donor concentrations, you might find it hard to pick up disappearance from the lumen. So one way to get around that is to measure plasma concentrations, because that will make it more sensitive. So again, correlation to human fraction absorbed, rank order is as expected. I have 5 compounds, and their P effective will appear similar to their fraction absorbed. So again, the technique works under controlled conditions. You can do other things, such as looking at site-specific permeability. In this case, the salt form of this base, the succinate, either formed a priori or in situ, has higher ileum permeability than jejunum. Those types of experiments can also be performed. You could look at prodrug activation. In this case, the prodrug essentially is converted to the drug form only in the upper GI, but not in the colon. You could also look at the sustained release feasibility, where if essentially your threshold is about 0.1, which is right around where metoprolol is, maybe this compound actually has a reasonable P effective or permeability profile all the way from the upper GI through the colon, so a sustained release may not be a bad idea. And finally, some quick comparison slides. This is the dynamic range, about 3 orders of magnitude for KCO2, For intestinal perfusion and excised tissue, it's about 1 order of magnitude. So KCO2 has a bigger dynamic range, makes it more sensitive, and in a second I'll show you how that can be used to your benefit. Variability, excised tissue, low to high, we see for the high permeability compounds about 25%, low about 40%. This is both between replicate and interdonor. Perfusion, not as great for compounds such as atenolol. Again, this is the limitations of the current non-absorbable water markers, or so-called non-absorbable water markers. KCO2, we average about 20 to 30%. Again, this is across passage numbers between days in culture and also different operators and pH conditions. So convergence of techniques. Convergence of techniques. So this is where multiple techniques working towards a common solution. The question being asked here was, you're contending with a hydrophilic nucleoside. How do I go about improving its permeation, because oral delivery is what you're going after. As a first pass screen, KCO2 may not be a bad idea, because you already know that you're looking at some permeation enhancers, maybe some ion pairing to try and get it across. But of course, KCO2 being a monolayer has some tolerability issues. So in the first pass, you recognize that maybe 3 of these excipients can move forward. A couple of other ones did not work as well. And of these 3, TPGS and Labrafil showed the biggest impact, because KCO2 is sensitive, can't pick it up. And then essentially you move forward to a more sophisticated technique, maybe excised tissue, because here your excipient concentrations can become more physiological, which can actually translate into your oral, solid oral dosage form. And finally, my conclusions. I looked at this in terms of economic impact, and Jack Cook, Barbara David, James Pauly put this together, I believe, in 2010, and they've done this a few times before. So for biowaivers, direct savings, you have indirect savings, which is your product getting to the market faster. There's also this redeployment or opportunity cost. So this is well understood. There are things that you can save along the pathway and also reduce the risk of unnecessary human exposure. In addition to that, there is also the consumer perspective, where you're trying to make medication cheaper for the patients and more available. And finally, as we discussed before, there are multiple avenues that an early classification becomes useful. And with that, maybe you set up a better development path which can focus on relevant assays, eliminate unnecessary in vivo testing, and hopefully add to your substantial time and cost economies. And that was my last slide. Thank you for your attention.